Hi. So, in previous session, we have studied the classical, alternative, and lectin pathway. These are very important complement pathway. Okay. Now, in this session, I will take you to the regulation of complement, because you can see that this complement needs to be tightly regulated, and there are some diseases associated with some complement. Okay. So, I will take you to all these things. Okay. So, let us begin with regulation of complement, and as you have learned that complement components are capable of attacking host cell, and because they can undergo spontaneous activation and inactivation. So, everything need to be tightly regulated. If it will undergo spontaneous inactivation, then microbe cannot be there will be a no formation of membrane attack complex, and then there will be an increase in number of microbe, and that result to the disease. Okay. If they are not stabilized with other component which is highly labile, then that will be also a problem. Okay. So, if you see very carefully the all three complement pathway, the C3 convertase is a major amplification step. Okay, and therefore this needs to tightly regulate it. Okay, if this C3 convertase is not tightly regulated, or if there is a dysregulation, that may result to the disease, or that may result to the um, growth of a microbial infection. Okay, whatever individual is infected, there will be a increase in number of those microbes. So, this regulation we basically we will discuss in two major component. One is soluble factor, which is regulating this complement. Here you can see there is a C1 inhibitor, okay, which is also denoted as a C1INH. Okay, and this inhibitor is basically here you can see it is interacting with C1R, C1S of classical pathway, if you remember, which is a subunit of C1, okay, and C1Q, okay, and it is also interacting with MASP2 of lectin pathway. Okay. So, how this works? Basically, it displaces the C1R or S, and which is in case of classical pathway, and in case of lectin pathway, MASP2 basically inhibiting this activation of MBL. Okay. So, this is a very important factor which can regulate the initiation of classical and lectin pathway. And if there is some defect in this C1INH or C1 inhibitor, then that will result to a very complicated and very rare hereditary disease, and this disease is known as hereditary angioedema. Okay. So, this disease is basically characterized by the individual basically frequently they develop swelling in any part of their body. Okay. And this swelling can be extremely fatal if this swelling is taking place in inappropriate or in some vital places. For example, if this swelling will take place in respiratory tract, then that will result to the choking of respiratory tract, and then the individual cannot breathe. Okay. And if it is taking place in the gut, then that will be also very complicated situation. Although this swelling, there is an episode of swelling, and this episode of swelling is resolving by its own, but If this swelling is taking place in, for example, respiratory tract or gut, then that may create a complication, okay, and it could be extremely fatal, okay. So, another molecule is C4 binding protein, which is also denoted as a C4BP, and this C4 binding protein is interacting with C4B, and basically it displaces C2A, a cofactor for C4B cleavage by factor I. So, factor I is basically a serine protease which cleaves the protein, okay. Another is CPN1, which is a carboxy peptidase N. This is a simple carboxy peptidase N, and this carboxy peptidase N, the name is very clearly suggesting that they basically cleave the C3A and C5A, and it basically make it inactivated, okay. Another is factor H, which is binding with C3B, and basically it displaces capital B small b. Capital B small b is a larger fragment of factor B, okay, and it is also cofactor for factor I. And its deficiency, the factor H deficiency, can result to the age related macular degeneration. So, what is age related macular degeneration? So, in vision there will be the straight vision, the central part will be the blurred, or it will be not visible. So, that is known as a macular or macular related disease, and if there will be a degeneration of macular in which is present on retina, then that will result to the age related macular degeneration. Another could be the atypical hemolytic uremic syndrome. So, this is also very rare disease. So, this in this disease there will be a formation of a small clots, okay, and this clot can go in any places, particularly in the kidneys, and then that will cause the variety of problem, okay. There is a factor I, which is basically factor I is interacting with C3B and C4B, okay. This is basically a serine proteases, and you can understand that C3B, which is a important constituent of C3 convertase, okay, and C4B also. So, this factor I basically cleaves these C3B and C4B, and if there will be a deficiency of factor I, then there will be a low level of C3, and that will also cause the hemolytic uremic syndrome. There is another molecule which we call it as a protein S. This protein S is basically interacting with C5b 6-7 complex, and basically it is involved in inhibition of MAC formation, okay. So, these are all the soluble factor which is regulating the complement. Another is the membrane bound factor which is regulating complement. The first is CRIG, okay, and this is basically binding with C3b and IC3B. This is inhibitory C3B and C3C, okay, and basically this their action is that inhibit the activation of alternative pathway. So, CRIG is playing very important role in alternative pathway, okay. So, if there is some problem in CRIG, then it will increase the susceptibility to the blood-borne infection. Another is these are the receptor, these are the complement receptor 1, which is also known as a CD35, which is a receptor for C3b and C4b, okay. So, basically this is a cofactor for I, these molecules, and it displaces the capital B small b, that is factor B larger fragment, from C3b, okay. So, this makes a C3 convertase, if you remember, and C4A and from C2A from C4B. Okay. Another is a decay accelerating factor or DEF or CD. It is also known as CD55. It is the ligand will be C3 convertase. Okay. It basically displaces the factor B larger fragment from C3 convertase and C2A from C3B and C4B. Okay, so, and if there is a deficiency in this DEF or CD55, that will result to the proximal nocturnal hemoglobinuria. Okay, so what is the meaning of this term? Proximal means sudden, nocturnal means night, and hemoglobinuria means there will be blood in the urine. Okay. So in these individuals, this is again a rare disease, but in these individuals what is happening, when they wake up in morning, then their urine will be having a blood, it will be a dark red color, it could be a brown. Okay. So they will produce lot of blood in the urine. Okay, or dead blood, which will turn to the brown in color. Okay. So this is also very complicated and rare disease, and this can there is some drug which I will discuss in subsequent slide, which can be used for this proximal nocturnal hemoglobinuria. Okay. So there is another membrane bound factor, that is membrane cofactor protein, that is MCP or CD46, which is interacting with C3B and C4B. Okay, and action will be cofactor for factor I, and if there is a deficiency of these factors, then that will result to the atypical hemolytic anemia. Okay. The another molecule is protectin, which is also known as CD59. It is interacting with C8, and basically it inhibits the MAC formation, and that causes the proximal nocturnal hemoglobinuria. Okay. So these are the regulatory factor, and you can understand if there is some problem in this factor, that result to the development of disease. Now I will talk about there is some microbial pathogen which evade this complement system. Here you can see that Neisseria meningitis, this can so basically this can there will be some invasion molecule which is a factor H binding protein. They make this factor H binding protein, these microbe, and interact with factor H and inactivate the inactivates bound C3b. Another is Borrelia burgdorferi. This is also pathogenic microbe, and they make a protein which we call it as a outer surface protein E. Okay, this also interact with factor H and inactivate the bound C3b. Streptococcus pneumoniae, this is also very pathogenic microbe, and they make a pneumococcal surface protein C, and this is also interacting with factor H and inactivates bound C3b. So here you can see that the microbe also have some strategy to evade the complement system. Okay. Some of these microbes secrete some protein like Neisseria meningitis. This makes a Pro-A, which is binding, which is like a C4 binding protein. Okay, if you remember the previous slide. So they make the C4 binding protein and inactivate bound C3b. Staphylococcus aureus, they have a clumping factor A, and this is basically binding with factor I and inactivate bound C3b. The Staphylococcus aureus also has another 3 proteins: Staphylococcus protein A, staphylokinase this also they make this protein, and complement inhibitor. So basically all these protein of Staphylococcus aureus can inactivate the bound form of C3b and bind to the FC region and interfere with C1 activation. They can cleave the immunoglobulin, they can inhibit the convertase activity. So in that way these pathogen can evade the complement system and establish the infection and cause disease. Now I will talk about the some complement deficiency, although I have discussed in previous slides, but I will sum up in separate session in this session with these slides. So there are some complement deficiency, and that will be result to the disease. Here you can see that the deficiency of C1q, C1r, C1s, C4 and C2, basically that results to the enhanced formation of immune complex, and this enhanced immune complex in the host result to the SLE like symptom, glomerulonephritis, vasculitis, and individual will have this recurrent infection of pyogenic bacteria. Okay, pyogenic means pus forming bacteria. Deficiency of factor D and properdin show that these individuals will be more susceptible to the Neisseria meningitis, but they are not susceptible to the another meningococcal bacteria. Deficiency of MBL, which is mannose binding lactin, a key component of lactin pathway, show increase in respiratory tract infection. Okay. Deficiency of C3 show frequent severe bacterial infection. Protectin, which inhibits the C9, okay, and therefore it inhibits the MAC formation, that is membrane attack complex. So the deficiency of protectin will result to the high risk of thrombosis. Okay. Anti-C5 is used for the treatment of I have told you proximal nocturnal hemoglobin urea. Okay, and so this there is a name of the drug is that eculizumab. Okay, so most likely this is a monoclonal antibody. Okay, this eculizumab is basically a monoclonal antibody against against the C5. Okay, and this is used for the treatment. I have already discussed this, that there is a deficiency of C1 inhibitor, and that causes the hereditary angioedema. Okay, I have explained you in more detail that these individual will develop a swelling in their body anywhere, and this is a spontaneous, and there is a it's not known how this swelling is triggered, and sometimes this swelling can result to fatality if it is taking place in vital places such as respiratory tract or in gut. Okay. So with this I am completing this complement, and now we will discuss another topic in subsequent session. Thank you.